Welcome back to this next video in which we are discussing coronary artery disease. Okay, so we're currently in the process of discussing coagulative necrosis, which is the form of necrosis that occurs in ischemic myocardium that is so badly ischemic that it's going to actually die and undergo necrosis. So let me just recap what we've said so far. So we've said that in this very ischemic myocardium, so ischemic that it's actually going to die, you're going to get a very severe lactic acidosis. So the lack of oxygen delivery to the myocardium will mean that anaerobic respiration will be turned on big style, and you'll produce lots of lactic acid, and that will lower the pH inside the cell. Okay, this lowering of the pH inside the cell is going to cause denaturation of loads of proteins inside the cell. In particular, it's going to denature the cytoskeletal proteins, and we've talked about how denaturing the cytoskeletal proteins is then going to cause membrane integrity disturbances, because remember, the membranes of the cell, whether we're talking about the plasma membrane or the membranes of organelles, those are just gloop. Okay, they're not structurally rigid at all. Okay, what keeps them in shape, what keeps them where they are, is the cytoskeleton, which links up with loads of proteins that are in those cell membranes. And if all of that's denaturing, then it can lead to disturbances in the cell membrane. For instance, we gave this example of it can lead to blebbing, it can lead to the formation of these blebs, which are these little diverticuli coming off the membranes, and here we've got it coming off the plasma membrane. And those membrane blebs can then pop, rupture, and of course that will spill the cytoplasm out into the extracellular fluid in the case of this bleb. So we're going to get the destruction of membrane integrity, and of course that's the start of necrosis, as we've discussed. So in coagulative necrosis, which occurs due to ischemia, it's slightly different to the other forms of necrosis, because you're not actually going to get complete autolysis of all of the cytoskeletal proteins, because actually when the lysosomal contents gets released due to the destruction of membrane integrity, a lot of those enzymes actually get denatured by the very low pH due to the lactic acidosis. Indeed, what happens is the cytoskeleton doesn't actually get completely destroyed. Instead, what happens is, if you look at these cardiomyocytes that have undergone this necrotic process and stain them with eosin, they stain bright red because the eosin binds very well to the denatured cytoskeletal proteins, and you can still see the outline of the cell, you can still see where all of the cytoskeleton was, so all of the shapes of the cells remain. You can't anymore see a cell membrane, which is why we often refer to them as ghosts of cells, because they no longer have a distinct boundary anymore. It's almost like a shadow of a cell, and they don't have a nucleus anymore. Firstly, because the nucleus underwent a pycnosis process where it shrinks down because of the low pH causing the DNA to aggregate together, and also because the enzymes in the lysosomes that break down nucleic acids, such as DNA, seem to be more resistant to the low pH than the ones that break down proteins, and therefore they survive and break down the DNA, so you get karyolysis after the pycnosis. Okay, so that's the process that these cardiomyocytes are going to go through then when they're exposed to this extreme ischemia. So just to give you a timeline then, let's say we start with the actual thrombosis forming inside our coronary blood vessel. So the acute coronary event actually happens, and then after that moment, a few seconds after that, the myocardium that was supplied by that now isn't getting perfused properly, that's going to start switching over to anaerobic respiration, and you'll start to get the lactic acidosis. The lactic acidosis will get worse and worse and worse, and if the ischemia is bad enough, then the lactic acidosis will build up to levels that it lowers the pH so much that you're actually going to start the coagulative necrosis process, and that generally occurs, if it's going to occur, within half an hour of the acute coronary event actually happening, within half an hour of the thrombus forming. Okay, so in the ischemic portion of myocardium that's going to undergo infarction, you're generally getting coagulative necrosis beginning after half an hour, and it will continue on and on and on until blood flow is restored. Now, before I actually talk about what happens later on once blood flow has been restored, because after blood flow has been restored, you're going to get the inflammatory process occurring, and I want to discuss that later on. Of course, we need to mention the fact that some people don't make it to that later stage. Some people die suddenly with a heart attack, and this is of course one of the reasons heart attack is so dangerous. It can cause sudden cardiac death. And what I want to do at the moment is explain to you why having a heart attack can lead to sudden cardiac death. And then we'll go on to the survivors, the people who don't suffer sudden cardiac death, and what's then going to happen in their hearts after that, and some of the long-term complications of having a myocardial infarction. Okay, so let me just get a new piece of paper up, and let's explain why some people can die suddenly with heart attacks. So why can having a heart attack cause sudden death? Okay, so the reason is that it can cause a fatal arrhythmia known as ventricular fibrillation. So I'm going to start off by defining the term arrhythmia, and I'll just make sure this is straight firstly. Okay, so let's start with the term arrhythmia, and then let's actually talk about what's going to happen in ventricular fibrillation. So an arrhythmia, it means that there is a problem with the cardiac rhythm. You can see that in this word, there is the word rhythm. So there is a problem with the cardiac rhythm. There is a problem with the cardiac cycle that the heart continuously goes through of atria contract, ventricles contract, atria contract, ventricles contract. And of course, that rhythm of the heart is orchestrated by a very complicated electrical signaling process that goes on in the heart. So really, what an arrhythmia is all about is a problem with the electrical signaling processes that occur within the heart. Okay, so whenever anyone talks about cardiac arrhythmias, it means something is going wrong with the electrical signaling processes in the heart, and often these are very pathological because they can then lead to the heart no longer undergoing the cardiac cycle correctly. And of course, if the heart doesn't undergo the cardiac cycle correctly, it's not going to pump blood potentially around the body correctly, and that's very, very important for life. Okay, so the reason then that heart attack can cause sudden death is it can result in a very dangerous arrhythmia known as ventricular fibrillation, and I want to explain what happens in ventricular fibrillation. Okay, so in ventricular fibrillation, the ventricles start contracting or start conducting electrical signals and being stimulated to contract at a ridiculously fast rate. So normally, the electrical signal that is going to cause the heart to beat is generated in the sinoatrial node, and usually the sinoatrial node throws off electrical signals at a rate of about 75 per minute. Okay, and these propagate down the entire heart and cause the cardiac cycle, cause a cardiac cycle. Okay? In ventricular fibrillation, what's going to start happening is the electrical signals are going to start being generated in either the left ventricle or the right ventricle, rather than up at the sinoatrial node. Okay, so let me just draw a picture of the heart, a simple picture of the heart. So I'm going to draw a very simple cross-section of the heart, like so. So we're going to imagine having chopped through the heart at a weird angle. So I'll just get back up my picture of the heart here, and I'll explain the angle we're going to chop through. So here's my original picture of the heart. We are effectively going to take a cross-section, a plane, through the heart. You can imagine taking your knife and putting it at this weird angle and chopping through, and the idea of putting it at this weird angle is so that you can see all four chambers of the heart. Okay, so hopefully you'll understand when I draw this picture what we've done. So here, this is of course the left atrium, here we have the right atrium, here we have the left ventricle, and here the right ventricle. And I think it might be worth just reminding ourselves of how the normal electrical signalling occurs within the heart, and then we'll contrast this to what happens in ventricular fibrillation. So, let's start by putting the sinoatrial node on here. So here, up in the right atrium, there is a specialized portion of cardiomyocytes known as the sinoatrial node, and these cells are capable of generating electrical signals spontaneously at a rate of about 75 per minute. So I'll just put SAN here for sinoatrial node. What will happen then is the electrical signal will be released from the sinoatrial node cells onto the surrounding cardiomyocytes, and it will spread from atrial cardiomyocyte to atrial cardiomyocyte. Now remember, it can't spread directly from the atria to the ventricles because there is an electrical insulation between the ventricular cardiomyocytes and the atrial cardiomyocytes, and this is of course known as the atrioventricular septum here, shown in green. Okay, so this is representing this electrical insulation, this fibrous connective tissue barrier that's going to stop the electrical signal propagating from atrial cardiomyocytes to ventricular cardiomyocytes. So this is the atrioventricular septum here. Okay, so the sinoatrial node then is throwing these electrical signals off at a rate of 75 per minute, and the electrical signal will propagate from atrial cardiomyocyte to atrial cardiomyocyte to atrial cardiomyocyte and will spread down the atrial walls, causing the right atrium and the left atrium to contract. There are specialized, or at least most people believe, it is widely believed that there are special conduction pathways within the atria that conduct the electrical signal. They're specialized cardiomyocytes for conduction, and they conduct it faster than the normal cardiomyocytes. So we believe that there is one that takes the electrical signal and very quickly delivers it onto the left atrial cardiomyocytes, and this is known as the interatrial pathway, also known as Bachmann's bundle. So I'll just write this here. So this is a special conduction pathway of cardiomyocytes to deliver the electrical signal from the sinoatrial node to the left atrial cardiomyocytes very quickly. Okay, because of course you want the electrical signal ideally to be spreading down the right atrium and the left atrium in synchrony. Okay, and of course the sinoatrial node being on one side, being on the right side, doesn't help with that. So we do need a conduction system to deliver the electrical signal there extremely quickly. In addition, we believe we have conduction pathways to take the electrical signal from the sinoatrial node to the one window through the atrioventricular septum. So the way the electrical signal propagates through from the atria to the ventricles is there is one window in the atrioventricular septum, known of course as the atrioventricular node. And I'm sorry I keep knocking the piece of paper. So this is the AVN here, the atrioventricular node. And this is a specialized bunch of conduction cardiomyocytes which can conduct the electrical signal from the atria to the ventricles. And we believe that we have specialized conduction pathways to take the electrical signal from the sinoatrial node to the atrioventricular node, an anterior one, a middle one, and a posterior one. So 3 internodal conduction pathways here. So they deliver the electrical signal from the sinoatrial node to the atrioventricular node, and meanwhile, as it's being delivered to the atrioventricular node, you're also getting it spreading along the atrial contractile cardiomyocytes, and those will contract when they receive the electrical signal. So you've had atrial contraction. The atrioventricular node takes its time in conducting the electrical signal from the atria to the ventricles, so that the atria have time to push the blood into the ventricles before the ventricles are going to contract. And then what's going to happen is it's going to deliver it onto the ventricular conduction system. So you have the bundle of His here, which splits into the right bundle branch, like so, and the left bundle branch, and these are special conduction pathways that are insulated from the surrounding contractile cardiomyocytes, and they are going to release the signal down at the bottom here at the apex of the heart rather than up at the top, because of course you want the wave of contraction to spread up from the bottom to the top so that you're pushing the blood out of the ventricles rather than down into the bottom of the ventricles. And then of course they end with Purkinje fibers, which are the fibers that actually deliver the electrical signals to the ventricular contractile cardiomyocytes. So I'll just mark those on in green there. So there's the normal way the electrical signal propagates through the heart, and just to complete it, the Purkinje fibers will give the electrical signal to the cardiomyocytes down here. It'll spread up the ventricular walls, and that will cause ventricular contraction. So that's the normal way the electrical signal is propagated up in the sinoatrial node and propagates through the entire of the heart and causes the normal cardiac cycle. Now, in ventricular fibrillation, what's going to happen is electrical signals are going to just be generated at an incredibly fast rate in the ventricles, and they're going to cause the ventricles to try to contract at a ridiculously high rate. So remember, the sinoatrial node usually generates electrical signals at about 75 per minute, so 75 beats per minute. Okay, in ventricular fibrillation, you can get electrical signals being generated in the ventricles at rates of up to 500 per minute. So the ventricles can end up contracting, beating at a rate of 500 per minute. Now that's a very extreme example, but it can get very, very high. And I'll explain what the problem with the ventricles contracting at that sort of a rate is in a moment. Let me firstly explain how this can end up happening because of a heart attack. So when you've got a portion of myocardium that is undergoing coagulative necrosis, it's going to stop functioning, and it's going to start having very aberrant functions. So let's say this is the portion of the ventricular wall. So I'm taking a bit of ventricular wall here, and we could imagine that it's maybe this portion of the ventricular wall here, and we're looking at it from the outside, we're now looking at it from here. Okay, so this portion of the ventricular wall is undergoing the ischemic necrosis, the coagulative necrosis process. So it's going to be starting to stop functioning. Its membranes are no longer intact. It's not really going to be conducting electrical signals anymore. So you've effectively got a great big lump of myocardium that is not going to be participating in the electrical signal. Now, what this firstly can do is it can give rise to what are known as reentrant loops. Okay, and let me explain what is meant by a reentrant loop. So a reentrant loop is a place where the electrical signal in the heart could potentially go round and round and round and continuously regenerate itself. And when electrical signals do this in the heart, it's known as circus rhythms. Okay, we say that you've got a circus rhythm operating around some reentrant loop. So let me just explain what's happened here. So when we've got this portion of myocardium that's no longer participating in the electrical signal, so the electrical signal can't spread to this area, what can happen is the cardiomyocytes around the edge, so if I draw now some cardiomyocytes around the edge here, okay, like so, so I'll draw a few cardiomyocytes around the edge, like so. Okay, these can potentially propagate an electrical signal all the way around. So let's say this one has got an electrical signal, and let's say it propagates it onto this one, and this one propagates it onto this one, and this one propagates it onto this one, and it spreads all the way around. And what if it got to this one, and by the time it gets to this one, the first one has recovered from its electrical signal, from its action potential, and can now conduct another one? Well, then this one can just give it back to there, and it can go round and round and round. And of course, when you've got this happening, not only will they be spreading it to each other in the loop, but they'll also be spreading it to all the other cardiomyocytes surrounding them. Okay, so it'll be propagating out, and it'll be propagating across the entire ventricles and causing the ventricles to contract. And you can imagine that this could therefore be spreading electrical signals to the entire ventricles at a ridiculously fast rate. So this is one of the things that potentially you can get established around an area of dying myocardium. Okay, so that's one of the reasons that you can get electrical signals being generated in the ventricles at ridiculous rates when you have a portion of ischemic myocardium, because you can get these reentrant loops forming, which can then give rise to circus rhythms. Okay, in addition, what you can actually get is what's known as ectopic foci. Okay, so these dying myocardial sites, when they're actually going through the process of dying, they can start, before they've actually completely died and their membrane is completely gone and they're not capable of conducting electrical signals whatsoever, when they're in the early processes of being damaged hugely by the ischemic process, then they can start dysfunctioning hugely, and they can start actually generating electrical signals spontaneously in the way the sinoatrial node cardiomyocytes would. So dying myocardiocytes can end up generating electrical signals spontaneously in the way that sinoatrial node cells can. The mechanisms for this are very badly understood, but it is an observed phenomenon that injured cardiomyocytes that are behaving that have very aberrant behaviour because of some sort of insult, in this case ischemia, can end up behaving in very strange ways and generating spontaneous electrical signals when they would have never been able to do so before. And such cardiomyocytes that are capable of doing this are known as ectopic foci. Ectopic, remember, means in the wrong place. Okay, so these are pacemaker cells, well, cells with pacemaker ability now, that are in the wrong place. And these can also then generate electrical signals which will propagate across the entire ventricle, both ventricles, and cause ventricular contraction. So both of these mechanisms, reentrant loop formation and ectopic foci, can lead to electrical signals being generated in the ventricles which will propagate across the ventricles and cause ventricular contraction at a ridiculous rate, far faster than the sinoatrial node would ever generate electrical signals, up to 500 beats per minute. Now, why is that such a problem if your ventricles start doing this? Well, at that sort of a rate, 500 per minute, you just don't have time to relax in between beats. So remember, normally what the heart has to do is it has to contract and then it has to relax and refill with blood. Contract and relax and refill with blood. And remember, the relaxation is a really important part. If you just contract and then never relax, then you don't actually perform the function of pumping blood around the body. And that's what's going to happen in this case. You're going to contract, and then you'll start to relax, and then the instant you start to contract, you'll just be signaled to contract again, and you will never relax back down, so you'll never get blood coming back into the ventricles. So instead what happens is basically the ventricles end up in a permanently contracted state, and they just sort of shudder, they shake, and these little shakes are them trying to relax, but before they can get anywhere with the relaxation process, they're then just forced to contract again because there's another electrical signal propagating along them. Okay, so what ends up happening in ventricular fibrillation is the ventricles end up permanently contracted, and they just shudder, which is them trying to relax, but then before they have relaxed, they're just being signaled to contract again, and they're contracting again, so they just sort of shudder and shake, and of course then they're not going to pump out much blood at all. Okay, so they have effectively become useless. They're not actually going to be pumping any blood around the body, and this leads to the phenomenon known as cardiac arrest. So cardiac arrest means when the heart isn't pumping any blood around the body, and of course once that happens, then the brain is going to die because it's not going to get any oxygen delivered to it, and you're therefore going to die. So that's how sudden death can result because of a heart attack, because you can get this fatal arrhythmia known as ventricular fibrillations, where you're just generating far too many electrical signals at a ridiculous rate, and the heart then just doesn't have time to relax between contractions and ends up not pumping any blood therefore around the body. It just sort of shudders and shakes in a permanently contracted state and ejects tiny little amounts of blood that are no use to anything into the major vessels. Okay, right, so that's the reason then that having a heart attack can lead to sudden death. So, what I now want to talk about is if you don't undergo cardiac arrest because of your heart attack, what will then happen afterwards? Okay, so I want to give you a timeline of what happens now. So, I'm going to start now with 12 hours. So we started obviously with 0 hours, when the acute coronary event had just happened, when you've just got the formation of the thrombus in the coronary artery over the ruptured atherosclerotic plaque. Then we had the fact that, ischemia was going to occur very quickly after that, and then anaerobic respiration was going to be turned on, and you were going to start getting lactic acid build-up very quickly, within seconds. And then by about half an hour, the coagulative necrosis process is starting to begin, and the cardiomyocytes are beginning to die. Now, the next time point I want to skip to is about 12 hours. And after about 12 hours, what will start to happen is either spontaneously or because of therapeutic reasons, and we'll go through later on the therapeutic reasons that this could happen, the thrombus will start to lyse. So the body has ways to get rid of blood clots, okay? One, because there is the potential, the body realises that there is the potential that blood clots will form in the middle of blood vessels and that these need to be lysed. In addition, of course, even if you do have a hemostatic plug, a blood clot that is in the side of a hole in a blood vessel, you don't want that to be there permanently. You do want to start retracting that back and replacing the blood vessel tissue, the blood vessel wall tissue that has been damaged and lost, rather than just having it replaced permanently with a hemostatic plug. Okay, so there are mechanisms for getting rid of blood clots, and after about 12 hours what will start to happen is the thrombus that is in the coronary artery will start to be lysed, okay, and therefore the blood vessel will start to reopen. You'll start to get blood returning to the ischemic portion of myocardium where the coagulative necrosis has occurred. So what's going to happen now? Well, of course, these cells have undergone the coagulative necrosis process. They've released all sorts of damage-associated molecular patterns into the extracellular fluid. Those damage-associated molecular patterns will have activated the sentinel cells' pattern recognition receptors, and those sentinel cells will be calling out to the endothelial cells with interleukin-1 and tumor necrosis factor alpha to bring in immune system cells. But now we finally have a delivery of immune system cells because we've got a blood supply again. Okay, so when the blood supply returns, you're now going to get the recruitment of immune system cells into the area. Okay, so at 12 hours, what's going to start happening is you're going to get neutrophils, polymorphonucleocytes, the main police, if you like, of the immune system. They're the ones that you bring in first. They're the least powerful, but you have a lot of them, and they can come in and deal with certain minor pathogens. Okay, so what's now going to happen then is I'll just draw a little picture of this. So how am I going to do this? I'll have this as a neutrophil. So here is a neutrophil coming in, and of course they're very distinguishable because they have the nucleus that has multiple lobes, and usually around 3, but it can be up to 5 or 6. So I'll draw one that has 4 next. Okay, here is a multi-lobe nucleus. Remember, they don't have multiple nuclei, it's just multiple lobe nuclei. So this is just one nucleus, it's just in a very weird shape. And now let me add on some of these cardiomyocytes that have undergone coagulative necrosis. So here is one of these ghosts of a cardiomyocyte here. Okay, then I'll have another one here, like so. And then we'll have another one here. Okay, so this is what the tissue will sort of look like after about 12 hours. You'll have a huge number of neutrophils coming in once the tissue is revascularized due to the lysis of the clot, either therapeutic or spontaneous. And they'll be amongst the cardiomyocytes that are still there with no nuclei, no cell membrane, but staining very well with eosin because of the denatured cytoskeleton still there. Okay, right, so that's what the cardiac muscle that has undergone this ischemic process, ischemic necrosis, is going to look like at 12 hours. Now let's skip ahead to what's going to happen within a few days. So starting at about 3 days, you're going to now start bringing in more powerful cells of the immune system. You're going to start bringing in macrophages, and macrophages are actually going to start clearing up this mess. Now, of course, this is only going to happen in your heart if you have survived the heart attack. Okay, if you've died of sudden cardiac death, then this isn't relevant. But if you survive the heart attack, you don't get a fatal ventricular fibrillation arrhythmia, then this is what's going to happen. After about 12 hours, you're going to get neutrophil invasion, and then after about 3 days, you're going to start getting more powerful members of the immune system being brought in, and I'll draw this on the same picture, which are macrophages. So what will happen is monocytes that are circulating in the bloodstream, they will be recruited by the endothelial cells, and when they come into the extracellular fluid, they'll then differentiate into macrophages, and macrophages are these huge, great blobs of cells here. Okay, right, so this is a macrophage here. Now, what do the macrophages do then? Well, the macrophages are going to clean up this mess. So at the moment we've got a huge number of dead cardiomyocytes, and we've got all of their cytoskeletal proteins just remaining here. Okay, and that now needs to be cleared away in order for the tissue to be replaced. Now, in the heart, unfortunately, and this is a big, big problem, in the heart it's not going to be replaced with new cardiomyocytes. The heart, despite the fact that it is one of the most important organs in the body, has a ridiculously poor capacity for regeneration. Once you have lost cardiomyocytes, they do not get replaced by new cardiomyocytes, unfortunately. You don't grow new cardiomyocytes. There's a huge amount of research in the stem cell field as to how potentially we can grow new cardiomyocytes artificially to replace the lost cardiomyocytes with, but it hasn't yet been used clinically. Okay, but your body is unfortunately not capable of replacing the cardiomyocytes that have been lost. Instead, what's going to happen is they're going to be replaced with scar tissue. What's going to happen is the macrophages, after they've cleared up this mess, are going to call for fibroblasts to migrate into the area. The fibroblasts will proliferate in the area, and they will start chucking out connective tissue. And you'll form something that's very, very strong to replace the cardiomyocytes. You'll form a beautifully strong connective tissue scar. The problem, of course, is that that's not capable of contracting like the previous cardiomyocytes were. It does the job of plugging the hole and keeping the heart intact, but it isn't capable of contracting, and also it's not capable of actually conducting conducting electrical signals. Okay, so let me describe what's going to happen then. So the macrophages start coming in after 3 days, and they are going to start dismantling the cytoskeleton. They're going to start breaking down these remnant cytoskeletons of cardiomyocytes. Okay, they're going to start clearing up this mess. And they're also going to start, as I say, calling in fibroblasts into the area. So here, this can be my picture of a fibroblast. So remember, fibroblasts are cells which synthesize a huge amount of connective tissue, such as collagen. Okay, so the macrophages will call for fibroblasts to come into the area. The fibroblasts will then start proliferating, and these will become the new major cell type in this area that we're going to have to replace because all of the cardiomyocytes are dead. And the fibroblasts, once they've got a nice population going, they're going to start chucking out connective tissue, and they're going to synthesize a beautifully strong extracellular matrix in the place of the cardiomyocytes. So overall then, by about 6 weeks after the heart attack, okay, so after 6 weeks, what you will have got is all of the cardiomyocytes that died in the heart attack, they will have completely gone. Okay, there'll be no remnants of them anymore. And instead, in the place where you had the heart attack, the ischemia occur, you will have lots of fibroblasts surrounded by a very dense extracellular matrix, a huge amount of fibrous tissue, and that's now known as a fibrous scar. So you form a scar in the place of the lost cardiomyocytes. So you do not replace the cardiomyocytes that you lost with new cardiomyocytes. So that's what happens if you survive your heart attack. After 6 weeks, you'll have a beautiful scar in the place where the heart cells have actually died. Now then, to finish this video, what I want to mention is what are the long-term complications of having a heart attack and surviving a heart attack. Well, the long-term complications, some of the long-term complications, are that it can still result in arrhythmias, because this scar tissue that you've got in that portion of myocardium, that has the same problem that we discussed up here. It's not capable of conducting electrical signals at all. Okay, so again, you have the potential to form reentrant loops around the scar tissue, and therefore you have always got that potential that circus rhythms might actually be able to form going around that scar tissue, and therefore that might be able to lead to an arrhythmia within the heart. Okay, right. So you do have the risk that you can develop an arrhythmia after a myocardial infarction because of the presence of that scar, which is disturbing the electrical signalling within the heart potentially. Okay, the other major thing that I will mention is that you have the possibility for it to develop into heart failure, and I'll just describe briefly what happens in heart failure. So heart failure means that the heart is going to fail the rest of the body. It's going to fail to do its job. Now, what is the job of the heart? Well, of course, the job of the heart is to pump blood around the body. Okay, so in heart failure, the heart is going to fail to pump an adequate amount of blood around the body. Now, why, after having a heart attack, are you at risk of then developing heart failure if you survive the heart attack? Well, because that portion of scar tissue, that's not capable of contracting. So let's say we've got a great big portion of scar tissue now in the wall of the left ventricle here. Okay, so you survived your heart attack, but you've now got a great big portion of scar tissue here, and let's make it even bigger. Let's have it all of this black portion here. Okay, that's all scar tissue now. That means that the actual ability of the left ventricular wall to generate contractile force is going to be hugely reduced, because now all of these cardiomyocytes down, a huge great chunk of your wall is now no longer going to contract. Okay, so the ability of this left ventricular wall to eject blood out of the left ventricle is going to be reduced. Okay, so this is going to lead to cardiac output potentially going down. And when cardiac output goes down chronically, what that can then lead to is the chronic activation of the renin-angiotensin-aldosterone system. Okay, so in heart failure, because of a weak heart due to the scar that you've now got in the wall of the left ventricle from the heart attack, cardiac output goes down chronically, and there is a system, an endocrine system for responding to cardiac output going down, and this is known as the renin-angiotensin system, often just abbreviated to the RAS. Okay, and I'll just write this out in full. So this stands for the renin-angiotensin angiotensin. Some people even add aldosterone system, renin-angiotensin-aldosterone system. Oops. So it would be RAAS in that case, but I think its proper name is just the renin-angiotensin system, and that should be an angiotensin, an N there, rather than angiotensio. Okay? So there is this system called the renin-angiotensin system which responds to cardiac output being low. Now, the renin-angiotensin system, it really is there to respond to hemorrhage, because usually the reason cardiac output goes down chronically is that you have had a bleed and lost a huge amount of blood volume. If you lose a lot of your blood, then of course your cardiac output is going to go down. So really that's what the renin-angiotensin system was designed to actually, or I don't know whether it was designed, of course no one knows whether it was designed, but that's really what the renin-angiotensin system is all about trying to do. It's about trying to bring back up blood volume in the case that you have got a hemorrhage. So this system now tries to bring up blood volume by reducing the amount of fluid that's going to be excreted by the kidneys and conserving fluid. So what that then leads to is far too high blood volume. Okay, so because this sphinx interprets cardiac output being too low as meaning that the blood volume is too low, it's going to raise blood volume through actions on the kidneys, and of course that's not a good idea because there's nothing wrong with blood volume, but it interprets cardiac output being too low as meaning that there is a problem with blood volume, not that there is a problem with the heart. Okay, it hasn't considered that possibility. Okay, so you end up raising blood volume, and that means that blood volume goes up beyond what it should be. So you end up with too much blood in your blood vessels, and this can then lead to fluid leaking out of the blood vessels because there's simply too much fluid in the cardiovascular system, and it can then accumulate in certain areas of the body. So for instance, the characteristic places for it to accumulate are in the legs, at the bottom of the legs, at the sort of ankles, so you can end up with swollen ankles. It can also accumulate in the lungs as well, known as pulmonary edema, and that can hugely affect your ability to exchange gases in the lungs. Okay, so blood volume going up leads to, for instance, pulmonary edema, which can lead to respiratory problems, so pulmonary edema. It also leads to angular or ankle edema, which more properly is called peripheral edema, so swelling at the ankles because fluid's just accumulating there because blood volume's too high. So peripheral edema. Edema just means the accumulation of fluid somewhere, and I'm spelling it the English way because I'm English. If you prefer the more sensible American spelling, just get rid of the O. Okay, so it can cause pulmonary edema and peripheral edema, and of course pulmonary edema is extremely dangerous potentially. In addition, what happens is the renin-angiotensin system being chronically on, which of course it does end up being chronically on because the cardiac output is chronically down, leads to changes in the structure of the heart. So this does seem to have some ability to affect the heart. It does realise that there potentially is a problem with the heart. And angiotensin 2, a molecule, a key molecule in the renin-angiotensin system, which will be chronically elevated if this is chronically activated, can affect the cardiomyocytes, and it can lead to bizarre changes in the cardiomyocytes that seem initially to be trying to make the heart stronger, but then overall, over time, lead to the heart becoming weaker. So this can also lead to what's known as cardiac hypertrophy, and cardiac hypertrophy is where all of the cardiomyocytes, when stimulated by the renin-angiotensin system's products, specifically angiotensin 2, so I'll just write down here angiotensin 2, when stimulated chronically by angiotensin 2, the cardiomyocytes can grow, they can undergo hypertrophy. So hypertrophy means a cell grows, so it might go from being like this to being like this. Okay, so the heart cells can end up getting bigger because of the chronic activation of the renin-angiotensin system, and it looks initially like a good idea because it's going to mean that the heart can become stronger again. It looks as though it's going to be an adaptive response. The problem is it's sort of like a response that nature hasn't actually got around to perfecting. Okay, the cardiomyocytes undergo this process, and initially it's fantastic. They are stronger, and then it deals with the problem temporarily. However, these large cardiomyocytes, they have big problems. They become very unwell. So initially they become fantastically strong, but then the health health declines, okay, and overall they become totally useless by the end of it. So what can happen is you can enter a vicious cycle of the heart just becoming weaker and weaker and weaker, because the renin-angiotensin system being chronically activated leads to pathological remodeling of the heart, this cardiac hypertrophy here, which doesn't seem to have been perfectly designed at all. Nature hasn't got around to perfecting it. It works short term, but then long term, these hypertrophy cardiomyocytes, they just lose the ability to contract altogether, and therefore the heart just becomes gradually weaker and weaker and weaker. It gets worse and worse and worse until the point that it just can't do its job at all, really, and that's the point that unless you get a heart transplant, you're not going to survive. Okay, so to summarise then, if you do survive a heart attack, you are at risk of developing arrhythmias, specifically circus rhythm-driven arrhythmias. In addition, you're at risk of developing heart failure because wherever the infarct has occurred, specifically if it's occurred in one of the ventricles, because those are the two important chambers of the heart, the atria are dispensable. If it occurs in the wall of one of the ventricles, and generally it does occur in the wall of one of the ventricles, what's going to happen is that ventricle's ability to push blood around is going to be hugely reduced, and that's going to lead to a reduction in cardiac output. Cardiac output going down chronically activates the renin-angiotensin system, which is a fantastic physiological system for dealing with hemorrhage. Okay, it interprets cardiac output being too low as meaning we have lost blood, and what it does is it acts on the kidneys and tells them to conserve fluid, so blood volume ends up going up and up and up, okay, far higher than it should be, because of course there was nothing wrong with blood volume, this was a problem on the part of the heart, and therefore you end up with blood forcing its way out of blood vessels and accumulating at the ankles, that's called peripheral edema, and also unfortunately accumulating in the lungs, which is extremely dangerous, that's called pulmonary edema. In addition, the chronic activation of the renin-angiotensin system leads to stimulation on the heart by one of the molecules of this system known as angiotensin II, which is aimed at trying to make the heart stronger again. It aims at trying to repair the damage, but it's not perfect. It doesn't work long term. So angiotensin II causes the cardiomyocytes to undergo a hypertrophy process. They become much bigger and they are initially much stronger, and you're thinking, great, fantastic, we've solved the problem. However, there are big problems with these large cardiomyocytes, and there is a lot of research. I used to work in a lab that was trying to find out the answer as to why these hypertrophied cardiomyocytes actually become so sick, why they lose the ability to contract altogether by the end of it, and it's believed to be big problems with the calcium signaling processes that are actually occurring inside of the cardiomyocytes. Okay, but something leads to these cardiomyocytes losing their ability to contract once they've undergone this hypertrophy process. In the long term, okay, in the short term, they gain ability to contract. They are stronger after it, but in the long term they become totally useless. And of course, the more cardiomyocytes that undergo this process, the weaker and weaker and weaker the heart goes. So the whole thing goes around in a vicious cycle, and the heart gradually gets weaker and weaker and weaker and weaker until the point that it actually fails to do its job properly. And then, of course, you are in mortal danger unless you get a heart transplant. So heart failure, chronic heart failure, is one of the things that can happen after having a heart attack. It's one of the long-term complications of having a heart attack. So it's all bad news, basically, after you've had a heart attack. It is a really, really bad thing, and that's why we strive so much in people with stable angina to stop them actually having a heart attack. Okay, so we'll have a break here, and in the next video what I want to talk about is biomarkers for someone having a myocardial infarction.